So, good morning, everybody. Now, today I'm going to talk about complex objects and distributions of objects. And the first thing, a little bit about, let's see, didn't work. So now it comes. So again, some books. I think you know them all by now. You can get them all online if you like. There will also be a couple of publications that I mention throughout today's lecture, which can be useful to take a look at if you want to dig deeper into the material. Here's also another list of papers that can be interesting to look at. I'm not really going to touch much of these things. It's more like an alternative way to look at these structures. Until now, we have been talking about pretty many things. We are actually, I think, beyond middle of the class. So we have been talking about image enhancement, reducing noise, enhancing structures, etc. Also understanding the histogram and how they can be used for segmentation. In particular, then going towards automatic methods. We did also look at component labeling, which is the next step. So the segmentation is only giving you the areas which correspond to some different, some specific characteristic, but it doesn't tell you about which region is separated from the other and doesn't give you a label on it. And that is what component labeling is doing. Today's lecture will actually take that one step further. It's the second part of today's lecture, actually. Then we did some shape analysis and also looked at some more complicated shapes, which are not maybe so easy to analyze with single metrics. So then we went into thickness maps. So today's lecture, a little bit about why and how we do measurements. Then I go into skeletons and watershed segmentation. The skeletons are minimal structures that still represent the topology. And watershed segmentation is the one that actually gives you labels for objects that touch each other, which the component labeling wasn't able to. So we have different environments. We can look at just the direct neighborhoods. Then there are different tools to do that. You can do Voronoi tessellation, which is in principle based on the distance transform, finding regions that are have local dominance to a center point. The other one is looking at the distribution tensor, looking at how things are distributed towards each other. You can also look at object alignment, how they are aligned towards each other. If you have a general direction in the material, or you have also that people, the objects are trying to avoid each other, or they are clustered in different ways, or oriented in different ways. The metrics we have, oh, actually, they come again here now. Let's look at what we want to look at. We want to look at different things in the images now, not only saying we have so and so many objects of that size, but we also want to look at what is the shape of them in terms of topology, or how they are organized relative to each other. And also how we can separate objects when they are connected. That's always a problem when you have all these bubble or grain images, also cells. When you have clusters of items, then you always have the problem that the component labeling will label probably large clusters together as a single one. And that is what we're going to look at, how to get around that today. And then we can also look at different shapes of objects and how they grow. that would be a question to answer. So what we have done until now is we looked at the different things. First, we had the image, which we could segment. Then we looked at previously at the distance maps of the foreground or the background. And this already gives us some information about the image, but it's still not easy to put that information into something that relates to the questions you want to ask. So we have to take it one step further now. And it's always important to find the right questions and put these questions into, and translate these questions into something that can be implemented as an image processing algorithm. So it could be, if you look at cells, you want to identify how many are alive. One thing could be the size, like small cells may be dead while larger cells are alive. So for doing that, we would look at the volume distribution. And I just see a little thing here. So now we got the arrows. It could also be that living cells are round, they are feeding well and happy, and dead cells are more spiky and pointy, so they are collapsed. Then we would have to look at the anisotropy of the items. Maybe you would also say that cells that are alive, we want to know where they are, these cells that are alive, or where they are most densely packed. We could visually inspect, of course. We could look at the color by color, it's by volume. Visual inspection is unfortunately a bit too qualitative, so we want to put really a number on it in this class. You can look at the positions of the items. What do they really tell us? Individual position probably doesn't tell very much, but if you relate it to distances between items, or maybe some source, then it starts getting more interesting. So saying that the positions, the items that are clustering towards, say, the source of food or source of water, that would give us more information. We could also make some boxes and count how many cells are inside each one. That would give you more a cluster analysis of where are the cells distributed and how many are there in these regions. And from that, we can go into comparing regions within the same sample, or even between two samples. If you have a time series, you could look at the same region with the same distance from the source of energy, and compare what happens over time when you start feeding more energy or taking out the energy. So that kind of analysis is what we want to do in the end. So what we still need is way to count how many we have. That was the problem before. As I said, the component labeling wasn't really able to handle that because you can't identify individual items if they are touching each other. You could also want to look at how many cells are in the closest neighborhood to the current one, and make a distribution of that, seeing how they are clustering together, how many are local, how many are more remote. And then also quantifying orientations. So for example, in some cases, if you have streamlines of some kind, could be channels that are oriented in some different direction, and you have items flowing in these channels. Want to look at how turbulent the flow is. So we need something that is adaptable, or at least a toolbox that helps us to answer a lot of questions. Also handling different types of structures, and that is what we are trying to aim towards now. The problem is, experimental-wise, mostly experiments are destructive, or at least altering the sample. So X-ray tomography, in the first claim, it's always said to be non-destructive. But it is actually ionizing radiation, so you need to give the sample some certain dose, and that dose can be lethal if you have living tissue. So we don't want to kill the cells, for example, by using X-rays. So we have to balance that in a way to not killing the items. Other methods, you have to cut and color the samples, and already this process is also destructive. In particular, if you do cutting, you may destroy some tubular structures or other structures, or maybe tilt them a little bit, and that can give you a biased information when you start analyzing the images. And then the next thing is, if you're doing time series or combined experiments with different modalities, then you also have a problem with the registration, that you have to align the samples between the images in a way that they actually show the same thing. And that can also introduce new artifacts. So you have most likely some kind of resampling, and that can destroy, or at least alter the structures you had from the beginning. So we need some good techniques that allow us to compare different samples of the same type, because usually we are going towards statistics and want to make comparison between different batches or over time or something like that. And come into being able to answer questions like, if the sample changed by something in one direction after treatment or over time. Could also be that the volume fracture fraction of some element is greater in the perimeter than in the middle, or if the organization of the items stay the same if you start provoking the sample. So there are many questions like this we want to answer in the final experiment we want to do, to get answers in our different scientific questions, scientific projects. So that was a little bit introduction to why and what we would like to have. And the first thing now, really tool that I'm going to talk about today, is a structure analysis. And skeletonization is a tool that is used a lot in many applications. Mostly it's about finding an abstract representation of thin structures. So it could be identifying roads by thin lines, could be following rivers or river deltas, also root structures. I have mentioned them a couple of times already. And in the medical application, we have blood vessels that also have very thin structures, and you want to analyze what they look like. And usually questions you want to look at is, for example, branching, length, also thickness distributions over the whole sample. And to do all this, we need some tool to reduce the structures to an abstract minimum. And another thing also, actually, I forgot to mention, are loops. Do the structures produce loops? Often in networks, in natural networks, you usually don't have loops. But if you go to, for example, road networks, then you can get loops like street blocks they are actually loops in the structure, and you can study how much looping you have in your images. You can also look at the tortuosity, how much a structure is wiggling around, if it's a straight line or if it's twisting a lot. So that is, for example, interesting for river analysis, also blood vessels and roots. Another thing is branching. And actually, all these things we're going to look at in the exercises also after today's lecture. And one example now that we're going to follow is from an EPFL dataset from the Computer Vision Lab. And anyway, in principle, just to look at what a skeleton could look like, this image is taken from Scikit image as an example. And this, of course, you can see that's a shadow image or contours of a horse. And if you reduce this horse to its minimum thinnest line, you will see the image that you have to the right. And from this, in theory, it should be possible to actually reconstruct and get back something that looks like the horse again. For that, you would need a combination with a distance map, where you would paint in disks, fill circles on the different locations. And it would actually probably get pretty close to what the horse looks like. Not exactly, but pretty close. So you have all the features, like if you look up here at the ears, they are here represented by forking. Also the legs, and here the tail is a little bit special, that it twists out into this kind of branching structure. But if you look at the tail part here, you can see that there are small gaps, and all these gaps, they need to be topologically preserved, and that's why the tail looks like this. So this is a very simple structure, and with simple, one thing is important to mention is that smooth contour lines make the skeleton very simple, and that's actually also confirmed by the tail part here, that when you start getting more cuts in the surface, then you also get more branching in the skeleton. So let's take a look at the street that I told you before from EPFL. Here you have an aerial image of a neighborhood with a street going through. And they were so kind to also provide a ground truth where the roads are that we want to identify. Now, in the next slides, I'm going to go through the routine, how we would do it through image processing with the tools that we have and will learn. First up, let's just look a little bit at the close-up. It was a little bit too much information in the first one. So what you can see here in this image is, one, as you have clearly the street, which is good. What can be a little bit disturbing are these trees, as you will see when we start segmenting the image. The color code right now is RGB, so red, green, and blue. And you can see that there are different colors. So we have the street, which is grayish. All the gardens, they're green. The house roofs, mostly gray. So that's almost the same as the street itself. And then there is something around here. Actually, I don't know what it is. It looks like some kind of scanning artifacts or something like that. So this is a quick qualitative analysis of this image. So let's try doing our first segmentation. And what I'm going to do here is now to convert it into HSV. So V is actually the intensity value. So, with that, we get a black and white or a grayscale image of the street. And I'm going to come back to that a little bit later, which kind of transforms you can use. Anyway, with this representation, we put a threshold on it, actually a fixed threshold. So it's just a very simple example, this one. So no magic using algorithms. And with that, we get some segmentation. You can see that the street is actually here, but you get a lot more, like the houses, probably the driveways into the houses. This particular is the artifact, which comes across here. You can also see that the trees, they're bulging in on this image. So it's not a very nice segmentation, but let's see what we can do with it. So, no. Oh, now I killed my data. Sorry, I need to go back. So there it is. So let's go back to the slide again. So we have our segmentation, and from this one, I want to continue doing something. Just again about this segmentation, here we did this color space transformation into HSV. We could also use something like a more automatic method, unsupervised k-means. In that case, I would actually combine all 3 color channels and possibly also the position. And with that, I would guess we will get much better segmentation than we got using putting only these thresholds. Otherwise, we could also start doing some supervised training with k-nearest neighbors, but that will require that we have a training and already marked up areas. Depending on the task and if what you're going to do, you could choose any of these or even going to more advanced methods. It's up to you what you would need, and also, again, how much time you want to spend on it. If it's a single image, it doesn't make sense to do a 3-week implementation of some fancy algorithm for something that could be done actually even with hand drawing within a few minutes. So it's a question about how well, how much data you have and what the purpose is of the data. But I would actually probably go for the k-means with the color, RGB colors. Oops, something happened. So there it is. Next step is to identify structures. And what we have here is, again, the image. We got what it's segmented, and now the next step would be to do labeling. Here I used the connected component labeling, and as you can see, we get different blocks. The colors coding is a bit confusing because some colors are repeated, but they are not connected, these objects. This you can also see if we look at the labels. This is a pretty much a downsampled image where you can see the label numbers inpainted on each pixel region. And you can see that this is 15 and this is 19, so it's really not the same. It's not easy to find a color map that is really representing the information well. So for example, if we would try here using the classic one, Viridis, it's really hard to see where the segments start and end because the gradient between 2 numbers is very small. So you need something else. And another one would be using a flag, for example. That one would probably give you a more interesting representation. So it's when you have all these labels, it's it's important to show us a color map that actually gives you the contrast between close numbered labels. Another way would be actually to just take random numbers, generate a random vector, and then you get really fantastic color mapping. So now we have these items and we want to reduce them into some minimal structure. And the first step would actually be to compute the distance transform. If you remember, the distance transform always produce like ridges, mountain ridges or something like that, if you take the landscape analog. And the skeleton is in a way represented by the ridge of these structures in the distance map. And that is actually what we are looking for. And some algorithms are actually using the distance transform to compute the skeleton. So let's compute the distance map of the largest component. So now I sorted out all the small details. We still have some things that are connected, which we maybe didn't want to, but anyway, we get the main straight structure. So looking at the distance map, you can see that there is a trend going along the midline of the straight. Sometimes when you have narrow throats like this, it's not so obvious, but you can anyway see that there is some kind of maximum line going through the whole structure. And this is what we want to grab now. And looking at that, grabbing it, one way would actually be to take a Laplacian filter and see what comes out. So convolving this distance map using this kernel that I have here. I can even make it centered. And see what comes out. And the result for a skeleton would be that we need to have not only the Laplacian, but we also need to have some criteria. And one way could be to look at a minimum distance. The intensity must be corresponding to a minimum distance, and the slope also needs to be fulfilling some criteria. And with that, the combination of these two, we can get our ridge. Still, it's probably not the most minimal ridge, but we are getting closer to a skeleton. And here you can see the distance map again, and Laplacian shows pretty nicely actually all these structures, how they are spanning out in the image. So you can see very faint here. Some structure going out to the edge, a little bit too much actually. And that is because, for example, you can see in this area, there is a pixel zigzag, and that actually creates a new branch in the skeleton. So this looks like something we can use. So now we just need to do some tracking along this. And in scikit-image or OpenCV, you can find a lot of different distance different skeleton algorithms. Here, this is the so-called medial axis, which is implemented already. It's even a little bit pruned because it doesn't produce all the spurious things that we saw in the Laplacian of the distance. But anyway, you can see that you get a lot of additional branches, except for the street that we wanted to get. But that is now for us to prune. And that's the next step to do. Another way of doing the skeleton is to do morphological thinning. That's a different approach where you do actually have this basic structure, and then you erode from the interface between object and the background, and then you erode until you can't erode anymore without breaking the topology of the item. And that's also one that is available in scikit-image. Here you can see some structures. First, we have the segmentation. Here, thinning was applied once, and you can see that structure has reduced. And now to get rid of unwanted structures, we applied here an opening, and you can see beautifully that the street is now more easily and more clearly extracted than it was on the previous one. And now we continue doing this and thinning until infinity, so until stability. And this is what we get. So it's almost the same as the previous one, but you can see looking back, the one based on medial axis actually produced a lot more unwanted structures. But I would say the reason why there are so many algorithms and ways to do skeletonization is that there are so many structures, and you probably have to try, if you want to do skeletonization, you have to try it out and see which algorithm actually works best for you. It's not given, actually. As usually with image processing, you have to find the correct recipe that is good for you. It may not be good for a colleague because they have some other problem, so you can't take exactly the same. The more similar the project is or the samples are, of course, the more same recipe you can use. But well, you have to make your own, explore with your own toolbox and see what you can do. But still, as you can see, this skeleton is still a little bit overgrown. You have still some branches that you don't want. And now we want to do some extra improvements on the data. So what we're going to do now is to identify where we have the junctions. So that is the points where a branch is going out from the stem. And a simple way of doing this is actually to use convolution again. And we use a simple uniform kernel, a 3x3, which is in principle the sum of all neighbors around the pixel. And that gives us this thick structure, which we actually don't want. We want the thinnest structure, so we have to mask out the skeleton from this one. And this is what we get. And looking closer to this one, you can see that yellow is the main branch pieces. So, and at the edges, you have a little bit different color. It's actually more visible on the thick one. So out here, you have blue, then you have yellow, and you have gray. And the gray corresponds to junctions. And that is a color which is higher. So in this case, you have, actually that's an interesting point. There is a gap in the middle. But anyway, a 4 means that you have 4 connections around. And then we have some interesting places down here, which is a little bit more difficult to analyze. But let's continue looking at this. Let's see, I need to go down. And now we get a little bit of a close-up. It's easier to see here. So in this area here, if you sum up normally, it's just a normal line, then you have a 3 as the sum of the neighborhood. But if you have a junction, then you're summing up actually 4 pixels, and that gives you the 4. And then you can see that there are a lot of junction places, and this one, well, that's this particular pixel is not a junction itself, but it's in a junction area, and that gives us this value. So this is a way to identify where the branches connect with each other, and that can be used now to break the skeleton up into smaller pieces. And looking at it, we have count 2, that is endpoints. So they are actually pretty useful. Then count 3 is all the edges between the endpoints and the junctions. Count 4, that are all our junctions. Count 5 is, well, a cluster of the skeletonization. So it's actually a kind of an outlier in the data. What we also can do is to use some smarter coding. This, what I showed just before, is only telling us we have a junction or not. And you saw some places where it was not clear exactly what kind of junction it was. And we can use a different coding. So here I have used a binary coding. This would produce very nice bit trains or bit sequences that can be analyzed, and each configuration gives a unique number. And that is interesting. You could actually tell the difference between if you have a junction going as a Y like this, or a junction going a Y downwards. You can determine the difference between them. And for some analysis, that can be interesting. And just look at how the analysis could be is that if you have x equal to 0, then it's clearly background. If x is less than 256, you have a pixel that touch the skeleton, but not really belong to the skeleton. And if it's something greater than 256, then it is on the skeleton. So already here we can do an easy selection. How to analyze this is, one is to actually look at the bit sequence and do analysis from that. You can see how many ups and downs you have. So switches from 0s to 1 or reverse. Or else you can also create lookup tables where you have some identified configurations. So for example, if you want to have a list of all Y junctions or all the X junctions, then you can separate those from each other, and particularly using this map with 8 connectivity. That's actually also one thing. The shape of the skeleton is depending on which connectivity you use with the structure elements in the thinning, and also when you do the reduction of the distance map. So the connectivity still plays a role. I think mostly people are using 8 connectivity, but if you want to speed up things, in particular in 3D, you may want to use smaller connectivity. So let's look at what these convolutions look like. We have a lot more colors in the 4 connectivity. You can see that we can distinguish top lines from bottom lines. We can do left, right, and then we have different numbers around each configuration. With the 8 connectivity, of course, the image gets more complex, but also we have more information in it. So depending on what you want to analyze, you can choose different kernels. Some cases, of course, you can just go for the plain convolution, and that's sufficient. And that's actually what we're going to do now. When we want to prune our skeleton, so it must be some, or ideally we want some model that defines that this can be a piece of the skeleton and this is not. We can use minimum branch length. So for example, all the little tiny branches, they are not relevant, so we skip them. And the other one would be to look at the branch width, and then we use the distance map to define this. So here is one I didn't execute. And looking at the segment length, we can see here Now, this is color coded with segment length. And we can see here that there are some lengths that are more represented than other ones. So we have one, the length 50 is very popular. Length 10 apparently also. And then in between, we have all the other stuff. And this was segment ID, sorry. In this one, we have the segment length coding, and you can see where we have the long pieces and where we have the short pieces. So this could be used as one criteria, saying everything with short pieces we don't want. The other one would be to look at here comes the ground truth. What we want to look at, we can see that this is the ground truth segmented by the people in EPFL, and here's our skeleton, and then is the question what we want to do with these segments. I would probably not use the segment length. That can be a little bit misleading, in particular if you have a lot of branching. So what we can look for instead is the maximum segment width. And no, didn't. Okay, I have to go back. Sorry for that. No, still not. Okay, I have to go further back. So this must be it. So now it should work. No, still not. Okay, that was not good. So actually, I would like to, let's see if the next one works or not. Yeah, the next one works. No, it's not a good one. So actually, it's 10 o'clock now. So I would say let's take a break from here. And then I find this problem and we continue in 15 minutes. So see you soon.